Hello, everyone. So we'll start the event. I will share the slide deck with everyone. Okay. So hello, everyone. Thank you for joining us today for our recent webinar around the Duplex Well, and that will enable enable the technology of membrane co-cultures, and will enable such applications such as Brain-Organoid-on-Chip and also Prevascularized skin models. So today we have two of our experts. We have Benoît Maisonneuve, who is our leader in biopharma and clinical translation projects. And we also have Alexandre Guichard, who is our product owner related to everything skin related and development of skin platforms. So just a quick summary of of what we're going to discuss today in our webinar. So first of all, we'll have Benoît joined by Eloise Castiglione, who will discuss the cerebral organoids with you guys. Then we'll have discussion around prevascularized skin, the proof of concept, with Alexandre Guichard and Thomas Bessy. And we'll finish with a quick Q&A session, and stay tuned until the end. We have a surprise for everyone. So just as a reminder, maybe some of you were there the last two webinars that we had, maybe some of you weren't there. In case you're interested, the last two webinars, there was one around Alzheimer's disease that we have shown with our collaborator ETAP Lab, that we're able to have an Alzheimer's disease co-culture model, and also we're able to show an injury model using A-beta oligomers. And the second webinar was around inflammatory bowel disease, where we were able to show that neurons were able to had an effect of immune cells on neuron cultures. And so if you're interested on either of the topics, I encourage you to watch the replays on our YouTube channel. And now we will focus on our topic of today. So just a quick reminder of what we have in NetTree, and maybe some of you already know our technology, maybe some of you are new. So what we propose is a microfluidic solution that is first high throughput and also interoperable. We have the advantage of our technology is that it enables compartmentalization. And as you can see in this picture, we have different microchips in a structure that has the same standard size as 1996 well plates. And the material that we use is, first of all, transparent. So you can use it with, you can do imaging. And also, it's a type of silicone that enables oxygen to enter, and you have to have oxygen exchanges with the environment. So it's a solution where you don't need additional pumps or other devices. And because it has the standard size of 96 well plates, it's compatible with a large variety of lab equipment. So the idea behind our technology is that we use neurons as biodigital sensors in the way where neurons will interact with different types of organs, and then we can have different types of phenotypic readouts that we can monitor in a dynamic and non-destructive way. And what we're striving for is to have, in the end, a digital library where we'll have predictive models of efficacy or toxicity, where we can compare in yellow, for example, a specific compound to different reference compounds in blue. And so today, what is the advantage with our technology is that today we know when we want to test different drugs, in vitro, you will usually mix different types of cells to have something that will be closer to a physiological model. So usually you will mix different types of cells, for example, neurons with endothelial or epithelial cells in a well. However, we know that anatomically there is natural compartmentalization. We have one that is the obvious one between the environment and the the interior, with the interface of the skin. You also have the compartmentalization between different types of organs, the vascular system, the organs, and between the different neurons. So what we have with our microchips on the right side, as you can see, we have our classic individual chip, which is the dual link, where you have 3 types of channels. You have the middle channel that is much smaller than the 2 adjacent ones. And so the liquid inside that channel is under a lot of pressure, which will mimic a barrier-like structure. And it will compartmentalize if you have 2 different cell cultures. For example, in the scheme on the right, where you have neurons in the 3rd channel on the right, and you have epithelial or endothelial cells, for example, in the left channel. And then you have microchannels that will help the neurons to innervate the cells that you have in the 1st channel. And so this is a classic innervation model. And what we have done is that we've gone further, and we have created a model, a device using a porous membrane, where you can see here on the bottom, we have a porous membrane that separates a bottom reservoir and a middle cubic well, where you can have 2D or 3D cell models. And so this new device is what we call the Duplex Well, that will be launched as a beta version in case you're interested in testing. And so here you have a close-up of what it will look like. So you have the neobento, you have usually 4 quarter bentos. In each quarter bento here you have up to 4 chips. So the unit of a chip, you have the middle well, where you can have your 2D or 3D cell models, with an inlet on the left and an outlet on the right, that are both connecting to a bottom channel. And the bottom channel is separated from the top well by a porous membrane. So here you have a deeper understanding of the specifics of this chip. Again, you have a bottom reservoir, and you have a middle well that is separated from the bottom by a porous membrane. The membrane has 400 nanometer pores, and the central well it has a volume of 142 microliters. So you can have different, for example, types of organoids that you can put in the middle well. And in this way, the membrane will have a role of a barrier between, for example, the bottom that can act as a vasculatory system and the top well. Here on the left, you have another way to look at the device, where you have the different components of the device. So you have the bottom perfusion channel, you have the membrane that separates the bottom from the top well, and you have the middle well, where you have the 2D or the 3D cell structure cultures. And on the bottom, for example, you can have different endothelial cells, for example. Here's another view, if you're interested, from the top of what the chips will look like. And so in case you're interested, we will launch a beta version of the Duplex Well, and either a light version where we have 8 chips, or a full version where we have 16 chips on the neobento. And now I will let Benoit, joined by Eloise, to present the Brain-Organoid-on-Chip.
Thank you, Nasiba, and thank you everybody for joining. It's my absolute pleasure today to talk to you about the work we've done on organoids in the Duplex Well. Just so for a bit of context, I guess most of you here today know, but in the last decade or so, organoid has appeared as a very innovative in vitro model. And that's the case for several organs, not just the brain. We focused our work on the brain first because that's the one where we had more interests at that moment, and also because of as you will see a bit later in the slides of the collaboration that is ongoing with Celtech. The big advantage of organoids is that they are three-dimensional self-organized cell structures, and they hold a lot of promises for a lot of different of applications. The big issue that there is at the moment with them is the lack of reproducibility and the difficulty to transfer this technology and scale it up to the industrial scale. What we propose here today is to combine the culture of organoids with the use of our devices in order to make this culture more reproducible and to get it ready to go to the industrial scale. The way we do it and what we are really relying upon, as you will see, is the fact that we have a very controlled fluid flow in the bottom channel. We can really not perturbate the cellular microenvironments that is around the organoids, and the fact that it's actually compatible with most industrial readouts and automation process is a big plus for us. So very quickly, just to give you a bit more of context as well. So as you know, especially for brain organoids, there are more and more applications that are appearing. It holds, as I mentioned before, a lot of potential, either for drug discovery, but also for neurotoxicity screening. And the sky is the limit, really, with organoids at the moment. So I don't know if, I guess most of you actually are aware with the beautiful papers from John Hopkins on organoid intelligence, with the fact that they use organoids and AI in order to try to train the organoids and try to actually get an answer question about cognition and behavior that hopefully will help us move more away further from the animal work. So without further ado, let's go a little bit more into it. So as I mentioned, the work has been done using the Duplex Well that is very efficient, in our opinion, in order to solve the drawbacks that I mentioned before about organoid culture, but also to try to bring it to the industrial scale. Here you have a quick picture. If you look at the left-hand side of your screen, at the top, you will see a picture taken from the top, actually, of a device with an organoid in it, and you have the organoid in immunostain organoid just below. So on the schematic in the middle, you can see there are other cells around, not just the organoids on each side of the membrane. Just forget about those for at the moment. We'll get back to that a bit later. Really what we are doing is culturing the organoids in the Duplex Well. So as Nasiba mentioned, the top well is an open well, so that is very convenient for 3D culture, and the bottom channel is a perfusion well. Sorry about that. So the membrane, the big point as well of the membrane is like it doesn't adhere, like the organoid doesn't adhere much to the membrane, which is quite convenient, of course. And also the flow of the media at the bottom helps to keep things moving. So in order to try that, the first big thing we did is, that's what I mentioned with Celtech, so Subbiotech from the CEA. That's the lab where Elo is currently doing a PhD. The first big step for us was to try to optimize the culture on chips in order to get reproducible cerebral organoids. So we tried a various array of conditions. The two main variables we played with were the time points at which the embryonic bodies were introduced in the devices, and three different ways of changing the media. So for the 3 time points, we chose what we call D plus 2, 11, and 18. And for the different way of removing the medium, we try by changing the media in the bottom channel, but also in the top channel every 2 days. We try to do, so the bottom one is always every 2 days, and really the difference between the conditions, it's about how we change the media in the top channel. So every 2 days for one condition, once a week for another condition, and not at all for the third condition. That enabled us to actually really see the impact of not doing any quick change of the microenvironment around the embryonic body first, and then around the organoid. So all the nutrients, all the different factors, they all, if you take the renewal strategy number 3, they will all take through diffusion. They will all come by diffusion through the membrane. And of course, we had some 24 well plates and 96 well plates control. So if we look at the shape of our organoids, so it's quite difficult to actually see just right here. But after 60 days of cultures, we had organoids work, like they worked for all the conditions. It's difficult to see from the pictures here, but actually they are much more repeatable in terms of size. You will see it in the graph on the next slide. Basically, in the duplex well, they look, as you would expect, quite round and no big surprises. If you look a bit more at the size distribution, what you can see is by doing the culture in duplex well, you tend to have much more higher reproducibility in terms of organoid size. So that's very important, because if you want to move toward, let's say, neurotox type of application, the way you will be able to characterize your organoid and make sure that they are ready to work and that you can rely on whatever predictive outcome you will have out of this is reliable. Otherwise, you won't be able to actually draw a conclusion. And when it comes to human health, it's very important to make sure that when we start, we got things running properly. If we look a little bit more at the situ architecture at 60 days, so it's something. It's a bit difficult to see here because it's very visual. But if you focus on the third condition, the one that is completely on your right, on the right-hand side of your screen, you will see that. So each picture that you have in each column and each array is a different organoid. Okay, they are not the same. And so if you look at the right-hand side, for example, and you take the third condition, you will see that they actually look a lot more alike than with any other conditions, especially a lot more alike when you compare it to the control in 24 well plates. What you also have, and that's very interesting, is you have a lot less cysts appearing, which is a pretty good thing when you think about organoid culture for once, but also you have rosettes in all of them with the expected morphologies. The patterns that are present along the organoid borders are well organized, and so it's a pretty good first step. We also looked at the transcript levels of the organoids via RT-qPCR for various genes, SOX2, TUB3, BCL2, and BAX. And what we found is that for all of the conditions, they were all within the same range than the controls. There's still some work that needs to be done on our side in order to try to go a bit deeper on this part and see if we actually have maybe better reproducibility in terms of expression levels and things like that. So just to conclude quickly on what I showed you up to now, so this Brain-Organoid-on-Chip, what we could see is that we could reproduce the expected morphology that has been published before. We have the right cell types within the organoids. The situ architecture are good and are reproducible, and the RNA expression levels are comparable to the one that you will have in the controls. The big two things that we are pushing forward at the moment are the fact that in terms of size and situ architectures, they are a lot more reproducible. And some work is still ongoing in order to go deeper and study them more in terms of cell types, of course, but also to go, as I mentioned, deeper in terms of various other expressions. As we could see, there are several moments when we can put the embryonic bodies within the device, and there are several methods in order to renew the media. You have a very quick, a very simplified matrix at the bottom left corner of your screen that shows the one that seems to be the most promising. Of course, as I mentioned before, and actually as you've seen before with the leader graph, you can definitely start to think about the next steps. And of course, one of them would be to go towards a blood-brain barrier. So that little drawing you had at the bottom, and you also had in one of the first slides when you can see cells on both sides of the barrier. So let's say, for example, on the top side, you could put some pericytes and astrocytes, and you could actually populate the bottom channel with some brain microvascular endothelial cells. All in all the work we did here showed that, yes, organoid is definitely a way to culture cells that holds a lot of potential, whether it's for drug screening or toxicological assessments. Heloise, for example, did a lot of work that will not be presented here, but has done a lot of work on neurotoxicity and screening actually the effect of compounds. And the fact that the organoids are more reproducible during the culture enables us to actually be able to screen about neurotoxicity during the culture, looking at the shape and, for example, and the evolution of the organoids during time. That's it for me. I will now let Alexandre take the lead.
So now the idea is to show you how we can use this chip for another application, which is skin and what we call prevascularized skin, and some data about proof of concept we did. First of all, a quick word about the context. Several in vitro skin models exist now, and they are usually reconstituting the different layers of the epidermis, even the dermis, by adding a fibroblastic layer. And these models are relevant for different applications, such as investigating epidermis cohesiveness or their differentiation, epidermis inflammation, or to test the efficacy and the toxicity of compounds. However, they often lack a compartment of vascularization, independent compartment of vascularization, which limits the transferability of the data to the clinic. So to address this limitation, we developed, among other applications, this device, the Duplex Well, to control the fluidic flows, to generate a vascular compartment independent with defined organization, and also to offer a ready-to-use technology compatible with standard methods, readouts, and standard lab equipment. Our chip models of irrigated or prevascularized skin allow a reproducible culture of cells, an access to high throughput thanks to our format, the SBS format, as explained before. Our Neobento, our format of chips, are compatible with automate or multi-pipette and so on, and some also readers. And the versatility of our architecture allows various applications, various cell culture, and also readouts. So we already explained you the architecture of this chip, but just now to show you how we can use it in dermocosmetic field. This chip, this device, has two compartments, as said before. On the top, the advantage of this top compartment is that it's open. Since it's open, it allows, for example, the addition of something from the outside, for example, exfoliant, or also it allows us to do hair liquid interface to, for example, recreate a reconstructed epidermis inside of the chip. And on the bottom side, we can culture cells such as endothelial cells on both sides to create a kind of big vessels. This is, I will show you some data about our preliminary data we have on this chip on this field. First, to show you that we use every cells we use in dermocosmetic are human, primary or IPS for some applications. So here for keratinocytes we use Promocell as a supplier. And we adapted their protocols in our different chips. So we have here two architecture, Dualink and Duplex Well. And in both architecture, the the keratinocytes grow well, they express the good marker, and they have a good viability. Same for endothelial cells, HDMEC. We use HDMEC coming from Promocell again, and we adapted their protocol to our devices. We observed the CD31 expression and the circular shape as usual. So cells grow well in our chip, and now when we put together cell, what's happening actually? We observe the standard morphology for both cells. As I said, we observed also the correct expression marker, standard marker. On the bottom side, you can see here an orthogonal view from confocal microscopy, showing that on the top of the chip you have the keratinocytes in red. On the bottom you have the endothelial cells in green, and so in between you have the membrane. So you can see here an overview of the chip. And interestingly, in our chip, when we grow cells together, they have different morphology and different arrangement and expression of different markers. For example, on the left side, you can see the keratinocytes grew alone on the left side, and when we put HDMEC, you can see that they aggregate together. So something happened. On the right side, you have HDMEC cells. Alone, you have a certain behavior and shape. And when you put keratinocytes on the top of them, you can see that it's more wavy, and the shape of the endothelial cells is different. So there is a cell-cell interaction, and this is a clue that there is a passage of molecule or something across the membrane from a compartment to another. So this is a good sign of functionality of the Duplex Well. And what are the perspectives and the evolution of this chip? The first one is to evolve this duplex in Duplex Well link. This is under development, and this chip consists on adding a third compartment here in dark blue to grow neurons. And this compartment is connected to the yellow one, to the skin, through microchannels in order to recreate, for example, innervated and prevascularized skin or other tissue. As said before, the open well allows also the air-liquid interface to, for example, reconstruct epidermis. And since our device are agnostics from the cells, we can imagine to put other epithelial, such as gut, scalp biopsy, for example, or nasal or gingival epithelium to recreate these other organs. Our chip, we are working on some devices, for example, and method to assess the permeability of the membrane. For example, the tear measurement tools or the dextran permeability. We are also, our device allows also the supernatant sampling to do ELISA and other enzymatic assays. And for the applications, our chips allows the application of a shear stress to mimic a flow. And for example, to activate endothelial cells, it's sometimes important and much more important in the blood-brain barrier application. We are also developing other connectors, small devices, to connect the chip together in order to recreate multi-organ. So we can imagine on our devices, we can have four Duplex Well or Duplex Well link in the same quarter, in the same area, and we can connect together thanks to this new jumpers connector we are developing. We can connect them together to create multi-organ and a loop. And these devices are also suitable for inflammatory analysis, since we are dealing with endothelial cells, with blood, with vascular cells. We can imagine also to put immunity and to follow inflammation or immune system. And if also thanks to the open well, we can, and if we use biopsy or explant, we can also apply topical product directly on biopsy, which is very interesting. And finally, we can also imagine to use this chip as a front cell-like, for example, to assess the transcutaneous penetration of product.
So I will take the lead on our last step of the webinar. So hopefully everything was clear, and now you know the different applications for our new technology, Duplex Well. So if you're interested in Duplex Well, you can test a beta version of Duplex Well that will be both in a light version with 8 chips in a neobento, or a full version of 16 chips in a neobento. And because we want to innovate further and to give you something even more complex, so as said previously, Alexandre, we have this system that we'll be launching during Q3 2024, which is the jumper. It's these little things that you cannot see on camera, but it's basically these little things, as you can see here on the bottom image in gray, which are types of connectors with 2 valves. Wait, I lost the presentation just for a second, which is basically a connector that will able to move fluids between different, for example, from one inlet to another, or between even between different types of chips. And what you can do even with the jumpers is that you have an automated system, which is the Seabird from Saitena, which is an automated system that will enable you to pump automatically. For example, you can put it directly into the Maestro, or with the connector and inside the plate. And you can, of course, connect different types of organs. And by Q4 2024, hopefully we'll be able also to have the same system in Duplex Well Link MEA, where you can connect different types of organs. For example, you can have chips with gut models connected to liver, to kidney models, and to have essentially more complex models if you want to study the interaction between different types of organs. So if you're interested in Duplex Well, you have a discount code. So you can contact us directly or check our web shop if you're interested in testing the Duplex Well device. So if you have any questions, you can directly all your questions in the chat. Okay, so we have a couple of questions that are coming our way. So we might start with some questions for Eloise. So we have questions such as, have you tested to maintain cerebral organoids for longer periods of time in Duplex Well?
Yes, sure. In the results that were presented...
Eloise, we cannot hear you. Or,
Is it okay? Do you hear me well?
Everything is fine.
Okay. So yes, to answer the question, yes, we have tested longer period. In the results that were presented, we cultured them for 2 months and analyzed at 2 months. But we also tried for 4 months of culture, so 120 days. And the analyses are still ongoing. But for now, we can say that for the morphology, all the organoids on the chip have the expected morphology and had during the 4 months, and are very similar to control organoids cultured in conventional support. So yes, it's possible to culture longer.
So there's another question for you, Eloise. And so how do you follow the growth of your organoids?
For the growth, it's based on pictures that we take of the organoids under microscope. And so by taking regular pictures during the culture at different time points, then we can easily measure the surface area of the organoids and plot against time and have the evolution. And this is possible because the chip material is optically transparent. So it's completely compatible with microscopy to observe the organoid directly in the duplex well.
Thank you, Eloise. We have a skin question. So we have some skin questions that are coming our way. Maybe for Alexandre, how are you sure that the cells are mature enough? How do you make sure of that?
Matureness is so actually here we are using primary cells, so they are not iPS cells, for example. So they are already differentiated, and for their functionality, we observed, as I said, we use immunostaining to assess the markers, to stain the marker, different marker. So for the keratinocytes, for example, we have the K10 and K14, which are markers of differentiation, and for endothelial cells, the CD31 are a marker of activated cells. Thomas, if you want to add more.
Yeah, well, we only, in the proof of concepts, we only used very simple models with non-differentiated skin, with just a layer of keratinocytes slowly beginning through the K10 expression. But we haven't gone through the whole process of measuring full skin in our devices yet.
Okay, and so were you able to grow other skin types or skin explants in the wells?
Other what? Skin explants. Yeah, so we didn't do it yet because, as I said, it's a really new architecture, a really new chip, and first we already did a co-culture of keratinocytes and endothelial cells. But yes, the size of the chip is the equivalent of a P96 well, the surface area. And so it should be suitable for skin for explants. For example, a 2 mm or 4 mm punch should be fit in the well. And for a reconstructed epidermis, actually, we didn't try yet, but we can by playing with the pressure, we can decrease and the level of the liquid in our inlet and outlet reservoir. We did a test, and we can decrease the level of the volume of the level of the medium in the well. So we know that we can dry the well to recreate the air-liquid interface. We observe it. But we didn't do it. Actually, we did once just for us to try. In one of our culture of keratinocytes, at the end of the experiment, we decreased the level of the medium, and we observed a kind of layer, different layer of keratinocytes, but we didn't do a confocal microscopy to assess, for example, or histology to assess the number of layers and the thickness, and or filagrine expression or so on. We didn't do that, but we are confident that it should work.
Also, because we made sure that all the different parameters of the membrane, for example, are good enough to fit through the membrane, and that everything has been sought for these applications.
Okay, so maybe if we go back to organoids, there was a question of which cell types are present in cerebral organoids. So maybe it's more of a question for Jessica.
Yes, thank you for this question. Concerning cortical organoids, they are mainly composed of neural progenitors, neurons, and astrocytes, and also a few oligonucleotides.
Okay, thank you. In skin-related questions, someone said that they found that our skin architecture was very interesting. Thank you. And the question was, do you know if the addition of endothelial cells affects the permeability of keratinocytes?
Maybe I can take this one.
Yeah.
Well, we have developed in-house a tier device to measure the transepithelial or the endothelial resistance that we wish to commercialize. And this device has highlighted that we can indeed measure the permeability of the barrier. We can see it increasing with the culture and also decreasing once treated with a molecule that will affect it. We, however, haven't tried on the co-culture yet. We expect that 2 layers of cells will increase the overall resistance because we can only measure the resistance through the 2 layers at once, but not individually once they are in co-culture. But it is something that we wish to measure indeed.
Thank you, Thomas. Someone is asking whether how to use our chip to study inflammation. Maybe Alexandre?
Yeah. So again, we don't do that yet. But for example, by using there is different strategy. Either using keratinocytes from donors, from patients with skin disease, skin inflammation, or by adding a cocktail of cytokine, for example, to induce, or pro-inflammatory cytokine to induce inflammation. And then the readout could be either the cytokine release by those by ELISA, for example, or also impairment of the membrane by microscopy. And we could also imagine so today we use a specific membrane. Actually, the membrane used in other devices and common in vitro. But we can also imagine to have a membrane with a bigger pore, for example, in order to assess the extravasation of cells, for example, if needed. So the chip could be modified if the application is relevant. So yeah, for information, Thomas, do you want to add something?
Okay, there's a general question around Duplex Well. Does the chip has MEA version? Not yet. So for now, we will have access to a beta version without the MEA for now, but it is something that might be interesting in the future.
And we have also IDs. We have another architecture with MEA, 2D chip, the Dualink. And actually, if you're interested about having a 3D culture, but also the MEA information, we have some ideas here to use a combination of chips to combine these 2 technologies. So contact us. We already think about this. Meanwhile, we are developing the Duplex MEA, but in the meantime, we have some ideas to reply to this question.
Yeah. So do not hesitate if you want more information to discuss this. We'll be happily to have a meeting and discuss this perspective with you guys. So one last question. Around organoids, maybe for Jessica or Eloise, is do you use Matrigel for your organoids?
Here we present only data performed using guided protocols, that mean without matrigel, but we have performed some preliminary tests with Lancaster protocols with matrigel. The device, the 3D cell culture is open, so it's possible to remove the organoid, embed it in matrigel, and maintain on culture on chip, so it's possible.
If I just may add on this, so while we did try and we did some work with matrigel, it's something we try to stay away from, mainly because of batch variations. There is a big issue with the reproducibility of matrigel in general, and so for a lot of preclinical application, especially if you think about application where the results will have to be submitted to an agency, that's a big no-go.
Okay, thank you guys. So hopefully everything was clear. In case you have other questions or you want to discuss further the Duplex Well, the application, do not hesitate to contact us. Remember, you have also a discount code if you want to test the beta version before the final launch. And we also have some incoming webinars. In June, we'll have a webinar around pain on MEA. And in September, we'll introduce our digital library. So stay tuned, everyone, and thank you for attending this webinar, and hopefully we'll see you in the next one. Bye, everyone.
Bye.
Thank you. Bye.
Thank you.